Hello all, today we are discussing sexual reproduction in Albuga. We have already discussed the vegetative structure and also the asexual reproduction in Albugo. So now we are discussing sexual reproduction in Albugo. Sexual reproduction in Albugo occur towards the end of the crop season of the host plant. We've already studied that fungus Albugo is a obligative parasite and it lives on a living host, and this sexual reproduction in this fungus occur towards the end of the crop season of the host plant. The development of sex organs and the onset of sexual reproduction is indicated by distortion, malformation or hypertrophy of the host plant. What is hypertrophy meaning? Meaning the stem or the leaf or the petiole of the plant becomes swollen up. Sexual reproduction is oogamous and involves gametangial contact. The male sex organs are antheridia and the female sex organs are called oogonia. Okay, so the antheridia and the oogonia, they are formed on separate but neighboring somatic hyphae, and the hyphae which bears the antheridia is called the male hyphae, and that which bears the oogonia is called the female hyphae. Okay, so what are the sex organs in Albugo? Antheridia and oogonia. Now let's look at the structure of each. Oogonium is globular multinucleated cell which is formed on the female hyphae. Okay, it is born terminally and is very rarely seen in the intercalary position. So where it is seen? At the tip of the hyphae. Okay, the terminal position. When it is young, the cytoplasm is vacuolated and the nuclei, vacuole, cell organelles and cell inclusions are uniformly distributed in the cytoplasm. As the oogonium matures, the nuclei undergo repeated mitotic division. So the oogonium is a globular structure. It is multinucleated, and where it is born? It is born terminally on the hyphae. Okay, so after the first mitotic division, the cytoplasm, it undergoes zonation. At first, we can see that it is completely filled with the cytoplasm, but later on it shows zonation. Cytoplasm, it gets differentiated into two regions. It forms the central dense mass which is called the ooplasm, and the peripheral region which is spongy, it is called the periplasm. Okay, so the cytoplasm of oogonia gets differentiated into how many regions? Two regions. The cell organelles are higher in periplasm, that is the spongy region. The cell organelles are high. Cellular organization and nuclear division occur in such a way that the mature ooplasm is uninucleate and periplasm is multinucleate. So this is the structure of oogonium. We can see that there is clear differentiation between the peripheral region and the central region. Now let's look at the structure of antheridium. The antheridium is an elongated multinucleated cell formed at the end of the male hyphae. Okay, that was also born terminally. It is cut off from the parental cell by a transverse septum. It has many nuclei, but out of it only one becomes functional, and it is paragynous. Okay, so this is elongated structure. It is multinucleated structure, and it is consist of many nuclei, but only one becomes functional. Okay, so this is the structure between antheridium and oogonium. Now let's look at the fertilization. The mature antheridium and the oogonium will come closely, that is, they will lie side by side. Okay, and at the point of contact, the wall layers become very delicate, and the oogonia, it gives out a receptive papilla, which is a small extension or a small outgrowth, which is called the receptive papilla. It later disappears. A slender tubular outgrowth is given out from the antheridia, which is called the fertilization tube. The content of the antheridium flows into the oogonium through this fertilization tube, and the tube comes in contact with the oogonia. Okay, so during fertilization, what happens? The oogonium and the antheridium come to lie side by side. The point of contact becomes very delicate, the oogonium puts out the receptive papilla and the antheridium puts out the fertilization tube, and then there is transfer of nuclei through the fertilization tube. A spherical and granular cytoplasmic body appears in the center of the oosphere. Now, this is called the xenocentrum. What is xenocentrum? It is a spherical and granular cytoplasmic body that appear at the center of the oosphere. The functional female nuclei migrate to it and remain attached to it. Meantime, the fertilization tube penetrates through the receptive spot in the oogonial wall and extend to the xenocentrum through the periplasma and the ooplasm. Its tip bursts and releases the content. Okay, so inside the oogonium, what happens? The spherical granular cytoplasmic body occur at the center of the oogonium. It is called the xenocentrum. It functions as the female nuclei. Okay, the functional female nuclei will migrate into the xenocentrum. The antheridial nuclei, which comes through the fertilization tube, it penetrates, it reaches the xenocentrum and it releases the content out there. The fertilization, now the fertilization is complete. Fertilization is now followed by plasmogamy and karyogamy, resulting in a diploid zygote. During this, only a single pair of nuclei remain functional and all the other nuclei degenerate. Okay, so during fertilization, only a single pair of nuclei, that is the male and the female nuclei, only a single pair will become functional and they develop into the zygote. Soon after the fertilization tube disintegrates, the xenocentrum disappears and the zygote remain within the oogonium. It secretes a thick wall around it and transforms to form the oospore. Okay, now after the fertilization, what happens? The fertilization tube disappears, the xenocentrum also disappears, and the zygote, which is inside the oogonium, will secrete a thick wall around it and it forms the oospore. Okay, now let us see the structure of oospore. The oospore has two walls, which is called the outer exospore and the inner endospore. Okay, now we know the nucleus of the oospore is diploid. It undergoes meiosis to form 4 haploid nucleus. These haploid nucleus again undergo repeated division to form in total 32 haploid nuclei. Okay, then what happens? This oospore undergo a period of rest after developing 32 haploid nuclei. So the oospore has how many wall layers? 2 wall layers. What are they? Outer exospore and inner endospore. Now what happens to the diploid nucleus of the oospore? It undergoes meiosis, first forming 4 haploid nucleus and then later it divides to form 32 haploid nucleus. Then the oospore, after all this nuclear division, it undergoes a period of rest. Okay, so have you understood up till here? Okay, if you have any doubts, please feel free to contact me. Okay, now let us discuss the what happens to this resting oospore. Now the oospore undergo germination under favorable conditions only. Numerous haploid daughter nuclei are formed, isn't it? Each nuclei gets surrounded by cytoplasm. It forms a reniform, uninucleated and biflagellate zoospore. So each nuclei, all these 32 nuclei, each nuclei gets surrounded by cytoplasm and each will transform into reniform. What is reniform? Kidney shaped. Okay, kidney shaped, uninucleated and biflagellated zoospores. The exospore crack open, the endosperm, from the endosperm a vesicle arises. First the zoospore migrate into these vesicles and then the vesicle ruptures and the oospores are released, or the zoospores are released from the oospore. The liberated zoospores gain access to the host and they come to rest. Okay, so after releasing, what does the zoospore do? It takes a period of rest. Okay, now have you understood the germination of oospore? Okay, now let's see what happens after the zoospore reaches the host cell. The zoospore now, after undergoing a period of rest, it withdraws its flagella. It rounds off and secretes a wall around it, and it forms a cyst. After some time, this encysted zoospore, it puts out a germ tube that gets into the host tissue through the stomata, grows inside the host tissue, and become a new uninucleate mycelium. Okay, its single nucleus elongates and undergo repeated mitotic division. First, it undergoes karyokinesis without the corresponding cytoplasmic cleavage. Hmm? Then the cytokinesis takes place. This results in haploid xenocytic mycelium. So what happens? The zoospore, after it withdraws its flagella, a wall layer is formed around the zoospore. It forms a cyst, and after that, after undergoing a period of rest, the zoospore then puts out a germ tube, and this germ tube grows into the host tissue, and it produces uninucleated mycelium. And each the nuclei, it undergoes division. First it undergoes karyokinesis, then it undergoes cytokinesis, and xenocytic mycelium, haploid xenocytic mycelium, is formed inside the host tissue. So this is the overall how the albugo sexually reproduces, how it produces zoospores, all this is the diagrammatic representation. You can see how the asexual reproduction, which consists of conidiophores, are also shown. Similarly, the sexual reproduction by the production of oogonium and antheridium, how the oogonium and antheridium come close to each other, how it produces fertilization tube, and how it develops into the zygote, and then how the zygote is it which is called the oosphere. No, it germinates, and then the zoospores are released, and how the zoospores get into the host tissue, how it forms the xenocytic haploid mycelium. This is the diagrammatic representation which is given here. Okay, so hope you all understood this portion. If you have any doubts, feel free to ask. Okay. Okay, thank you very much.